Now we will discuss nature of enzymes. If we talk about enzymes and their chemistry, they are always linked with protein because they always contain a protein. They might require other compounds or complexes that would make them functional. These compounds are known as prosthetic groups, or coenzymes is also another term that is used for these prosthetic groups that make these enzymes functional. They are normally vitamins, especially B vitamins, and that is the reason that vitamin B are normally implicated in our life processes to release energy from the food. Uh, so this is how these prosthetics group help enzymes to function their normal routine functions that they are designed to perform within the living cells. Some of the enzymes also need another component, which are metals. So these metals are also needed for these enzymes to function. For example, copper is one of the metal that is needed by many enzymes. So these metals are also essential, and that is why these should be provided in trace amounts from the food that we consume. So these can be proteins or protein with a prosthetic group or metal. These are additional things that are needed for the enzyme function within living cells. Enzymes, as I earlier said, that they would be breaking down the complex structure in our food, such as carbohydrates, fats, proteins, into simpler structure or simpler nutrients such as glucose or amino acids or fatty acids, and then these are utilized by human cells for growth, repair, and normal functioning. So that's why, in order to digest these foods and make them useful for living functions, enzymes are very critical. If you look at the carbohydrates, they are, as you know, they are the main source of energy from our food. These carbohydrates are broken down into simpler sugars, and these sugars are utilized, especially in the form of glucose. These are utilized as a source of energy for all of the functions that we perform, or all the energy requirements of living cells. So once there's an excess amount of this glucose, that may be converted into fat, and then this would be stored as a reserved form of energy. And once we don't get any carbohydrate in our food, and we need energy, these fats are broken down, because fats, whatever we store in our bodies, they are reserved form of energy. And when the food is not provided, these fats would be broken down by the addition of a water molecule that would be splitting these fats into simpler structure such as glycerol and fatty acids, and then they would be they become source of energy for the routine functions. The fatty acids that come from these fats, they would be converted into acetates, and these acetates are then utilized as source of energy. And whenever we have excess of these acetates, they again they can be converted back into fats. So this is how a reserve mechanism of energy release is in place in the living cells, especially in humans as well. Similarly, proteins can be broken down to amino acids. These amino acids, as we know that these amino acids are part of the protoplasm or the cell membrane or the cell structure, so they would be helping us in growth and repair. They would be deposited in the form of protoplasm. But in case when we are not left with any carbohydrates or fats in our body, these protein can also be oxidized to release energy, to fulfill the energy requirements of living organisms. These enzymes would always they contain proteins, and as we know that proteins are all made up of amino acids. Naturally, there are 20 to 22 amino acids predominantly there in nature, and their unique sequence makes a unique protein, and these proteins would have their own functions depending upon this sequence, and they would have their own identity basically. And this identity or this unique sequence again would make up chains, and these chains can interact with the side chains and the other chains to make up bigger structures. These are the tertiary structure or quaternary structure that tells us about the shape of the protein. And as we discussed that these shapes are important in case of functions of enzymes like the key and lock mechanisms. So these shapes are also important in the sense that they help in the regulatory process of our metabolism. If we talk about proteins, carbohydrates, there might be some other compounds such as phospholipids. They can be attached to the enzymes as well. They can make up different sequence of amino acids. These shapes, as I earlier said, they have unique functions. They would be providing us physical and chemical energy. The ideal temperature, if we talk about enzymes for their function, the range is from 15 to 65 degrees C. This is a range at which most of the enzymes would function. But again, there are examples, there can be exception that some of the enzyme might be able to function below 15 degrees C or above 65 degrees C as well. For example, there may be enzymes working above 71 degrees C as well. Otherwise, these enzymes mostly at the temperature range of 71 to 93, they would be inactivated. They would not be functioning their normal function at this temperature range, because higher temperature would inactivate many of the enzymes. And this phenomena is also sometimes used for good purposes in food preservation. For example, we can stop certain enzymes by heating food, and they would not be breaking down further food into simpler compounds, and the food would have longer shelf life. So if we talk about these exception, as we were earlier discussing, that there might be exception of some of the enzymes that can keep working at extreme temperatures, lower or higher. One of the enzyme that is present in fatty fish that has been shown to be still functioning at 100 degrees C temperature for 20 minutes. So 100 C is quite a boiling temperature, and that is a quite high temperature that would be basically active, and it would be breaking down fatty acids into phospholipids in the fish, and that would actually be making fish less palatable or less appealing, because it would be breaking down many structures within the intact muscles or tissues. Similarly, as we know that the temperature has an effect, pH has also an effect, and there has to be a suitable pH for enzymes to function. So this was a little about nature of enzymes in the living cells.